Well, good morning. So what did I start out with last week? Do you remember?
Homeostasis.
Come on. Up and down.
Piston.
The piston! What was the whole purpose of that?
Has a specific design.
As a specific design. So we have homeostasis, we talked about that. There's another concept which we'll get to down the road a little bit more, and that's called irreducible complexity. Have you ever heard that phrase? Irreducible complexity is just the idea of this. You can take something like a watch. Now this watch looks pretty simple, right? Remember the old days of the mechanical watches with the little flywheel inside? And on the outside it would look simple, but as you begin to tear it apart, you find all of these subparts to it, right? And typically if you remove any one of those subparts, the whole thing is junk. So you could take an engine, like the piston and the flywheel, all that stuff that we went through before, and usually if you remove almost any single part, will the whole thing fail? Yeah, you get no radiator in your car, and the engine's going to run fine for about 3 minutes, and then you're going to have nothingness. So remember that, especially as we get into the cell today. And the cells that we're going to look at typically run in the neighborhood of about 1/1000th of an inch in diameter. That's pretty tiny. And they're amazingly complex. So when you come in each time, I have the notes there, and as you walk in, on the left are the old ones, and as you move to the right, they're newer. So I put them out just in case you missed a particular Sunday. So remember the parts to homeostasis. The receptor, the control center, and the effector. And what we did is we talked about the thermostat. It tells you what the temperature is. If it gets too cold, then the furnace kicks in the winter, the air conditioner kicks in the summer, and it brings you back to where you have to be. Because we have a fairly narrow range for every cell, every organ system, and for you as an organism to live. If you go outside of that range, what will happen to you or your cells? You will die, right? So all of these homeostatic mechanisms, is this engineering at its finest?
Yeah.
How did all those systems evolve? Answer, absolutely impossible. And as we go through this, I'll give you a couple of really simple ideas that you can write down. And when somebody says, you believe in creation? Yeah. And then you can say, here's a very simple way to disprove evolution. So I'll give you a couple of those as we go along. So this is the thermostat that we talked about. Here's the idea of keeping your temperature as an organism within that normal narrow range of about 98.6. Then we can get to this guy. Uh-oh, this is beginning to look a little intense. This is the cardiovascular system. Now, we're not going to go through this piece by piece, thank goodness. And trust me, this is a simple diagram of the homeostatic mechanism. If you want to borrow any of my A&P books, I'll be happy to bring them in, and they have big flow charts like this for every system in the body. So basically your heart's sitting here, right? And it does this little pump, 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 correct? Now, is the heart in charge? No, it's got this little group of cells up here called the SA node, and the SA node, sinoatrial node, that precipitates electrical impulses which causes the heart to contract. So the upper chambers contract together, and then there's a little delay, and the lower chambers contract together. So here's that SA node. It has wires, neurons, that send signals to the atria, the upper chambers. Does that make sense? So they contract together. And then a few tenths of a second later, the lower chambers contract together. So I'm going to throw a lot of numbers at you. Remember when we were talking about your nerves, the neurons? Those are the cells that conduct electrical signals. They carry electric signals up to 120 meters per second. That's fast, right? Now, if those electrical signals are leaving that SA node at 120 meters per second, the upper and lower chambers will contract at the same time, right? So God evolved, God designed special neurons that slow the electrical signal so the upper chambers contract together at the same time, and then there's a delay and the lower chambers contract. Do you get that engineering? Can we explain that through evolution? No way, Jose. So, is the SA node in charge? No. You got the cardiac control center sitting up in your brain, and the cardiac control center has receptors throughout the body. It monitors temperature, it monitors blood pressure, it monitors different chemicals, and all of that stuff feeds to this little computer sitting right back in here, which has two nerves that come down to that SA node. One is the accelerator, and one is the inhibitory nerve. And depending which signals come down, that either increases or decreases the SA node activity to either increase heart rate and heart strength or decrease heart rate and heart strength. Do you kind of get that? That's what this is saying. Along with that, it can cause your blood vessels to get bigger, dilate, or constrict. So if you were to cut your arm and start bleeding, then your blood vessels will constrict so you don't bleed out as quickly and go into shock. You have all of these amazing systems. Do you kind of get that, folks? That we won't test on this one. So that is in a nutshell a homeostatic mechanism just for the cardiovascular system. Okay, so feel free to ask questions whenever you want. The cell. You started with one cell. There was a sperm and there was an egg. 1 + 1 equals, sorry to do the math trick here on you, buddy, but biology rules. So 1 + 1 equals 1, the zygote. And when you have a zygote, that's what you started with, one cell. And that eventually grew into the blob that you are of 100 trillion cells. You've got about 260 different varieties. Does anybody explain how all of that occurred by evolution? Can. Okay, so here we go. And I'm going to keep going over these parts to kind of get this in your mind. If we look at the cell, we have three major parts. The cell membrane sits on the outside, we'll talk about that, and it also has three main parts. Then you have the cytoplasm, that's the goo, and then you have the nucleus. Now, in Darwin's day, in the 1800s, they kind of knew basically about that stuff. They knew that there was this goo stuff, and they could see this little dark spot in the middle of the cell. So they knew there was a nucleus, but they had no idea what was in it, and we'll talk about what's in it. And they knew that there was this goo stuff. We used to call it protoplasm. Ever heard that term? Those of you that were alive in the early 1800s. And then around it is this thing called the cell membrane. So those are the three major parts. So, very basic drawing. What you have here is you have this cell membrane that wraps around, you have the nucleus inside, and then all of this space in between, that's that gooey stuff called the oh, you guys are smarter than you look. Very good. Cytoplasm. You see when my students don't like me? Yeah. Okay, I insulted them a lot. So, the cell membrane. Now that little, Renee, is your husband kind of tough? Is he a tough man? Yeah. Let me just say, most of him is a wimp. Just saying. And here's why. As I look at Peter, I'm basically looking at death, right? Because everything that I see as I look at him is dead. Because all I can see is his skin. Is the outer layer of his skin dead? Yes. You have 5 layers to your skin, and the outer layer of the skin is 28 layers thick. And it's basically almost all dead cells. So you're not too bad looking for being dead. So how amazing is your skin? Seriously.
Yeah.
It is phenomenal because it replaces itself, right? It can take all sorts of abuse. You work it, it get calluses. Some of you know what those are, others get real. Okay, so as I'm looking at him, his skin is pretty tough. But if I peel away his skin, I get down to live cells. How tough are Peter the Builder, how tough are his cells, his live cells? Absolutely wimpy. That's why they're enclosed in this sac we call the skin. Do you follow me? Because our live cells are phenomenally wimpy. That's why we don't do well with blunt force trauma, right? You get in a car accident or some crushing injury, and those cells don't stand a chance. Now your bones do pretty well, but your cells don't do well because they get crushed. So, cell membrane, 3 parts. Phospholipid bilayer, cholesterol molecules. You have to have cholesterol, it makes the cell membrane a little tougher. And then you have these proteins. So we're going to talk briefly about each one of these, but you got to get the idea of the cell. A couple, 3 weeks ago, I mentioned the idea that I would say and stand behind that your cells are more complex than Chicago, New York City, Tokyo, you make any major city, and your cells are much more complex than that. Phenomenally complex. We had no idea at all of this in Darwin's day. So how many parts to the cell? 3. What's the outer part called? Cell membrane. How many parts of the cell membrane? 3. Phospholipid bilayer, cholesterol, and protein molecules. So here's the cell membrane, again, a very basic schematic. So, do you see this little thing that has the circle and the 2 legs? That's a lipid molecule. And then you see this little thing here, that's the mirror image of it? That's another one. So you got this and this. And these are phospholipids, that's the name of the chemical, and there's 2 of them, so we call it a what? Phospholipid bilayer. Do you catch that? So this is outside the cell, this is inside the cell. Again, remember your cells average less than 1/1000th of an inch in diameter. So you have this guy and this guy, right? You with me? So that is what are those molecules called? Phospholipids. How many are there? 2. So this whole thing from there to there is the phospholipid bilayer.
Why don't they call it biphospholipid layer?
Quiet math guy.
The 2 seems misplaced to me.
Well, have you ever talked much with a chemist? Are chemistry people weird?
Yes.
I rest my case. Okay. No, seriously, a lot of when we name stuff in the world of chemistry, there's multiple ways to name it. Okay. And so you could do that if that's going to help you sleep tonight.
Okay, well, the reason I ask is...
Don't invite him next week.
To me, it sounds like there's 2 layers rather than 1 layer. Instead of... Yeah, maybe I'm just confusing myself.
Yeah, you are. You are. Okay. So, now, from there to there, that is your cell membrane, right? How thick is it? It's about 3 to 4 millionths of an inch thick. That's what's protecting your cell. Do you get it? Is fatty material very tough? No. So this, from there to there, 3 to 4 millionths of an inch thick, is basically 2 thin layers of oil. If I were to take a bucket, a 5-gallon bucket, and put in about 8 or 10 drops of oil, the oil's going to come to the surface and spread out real thin, right? You know what that is. I can take my finger, I can go through that, and then when I pull my finger out, the oil will fill back in. Does that make sense? That's basically your cell membrane. That's it. So that's all the tougher Peter is underneath. He's a wimp. Don't call him that at home, Renee. Okay. Okay, you get it? Now, out here, it's basically water. In here, it's basically water. Is this environment different than that environment? Yes. What keeps those two environments separate and different? The phospholipid bilayer. So a lot of the energy that your cell expends is either bringing stuff in or getting stuff out to keep the inside the same, which we call homeostasis. All protected by 2 thin layers of oil called the phospholipid bilayer. Now, it's really wimpy, so yeah, we have some cholesterol molecules in here that make that cell membrane a little tougher. By the way, if you eat too much cholesterol, can you get too much cholesterol in your cell membranes? Yeah, then they get stiff, which is a primary sign of aging. See how that works? No charge for the health stuff. We just throw that in. So what were the 3 parts of the cell membrane? Phospholipid, bilayer, cholesterol, which makes the cell membrane a little tougher, and then all of these glorious little things called proteins. Got it? How thick's the cell membrane? 3 to 4 millionths of an inch thick. Okay, so here we go. There's the phospholipid molecule. Do you see it? And down here, there's the phospholipid molecule. From there to there, 3 to 4 millionths of an inch thick. And that's just how it is. So literally, you could have something small go through that cell membrane, and then if you pull it out, those things will move back in and fill in that mini hole. But if you make the hole too big, then you lose, what's the H word? Homeostasis, and the cell will die. And if too many cells die, what do? You die. Do you get it, folks? Okay. Proteins. Oh my. So here we go. Here's my phospholipid molecules again, right? And you got some cholesterol. And then you have all these really nasty names for proteins that we're not going to go into, but they serve a whole bunch of different purposes. And we'll look briefly at those. So, proteins. You have receptor proteins. You have channel proteins. You have gated channel proteins, transport proteins, and we'll look at what basically some of those functions are. But again, oops, sorry. Do you see your phospholipid bilayer here again? Everybody with me? Very, very thin. Protein types. I want to get into this because proteins are so absolutely, to me, they're fascinating. Anchor proteins. These anchor the membrane to the cytoskeleton. Now, what we're going to see when in a future picture, we're going to see inside the cell, you have this matrix of tubes and strings. And those tubes and strings are made of proteins, and they actually make a skeleton inside the cell. The cell is so wimpy, it would collapse on itself and they would all be flat. But you have this little, and again, none of this was even known until about the 1940s, because they couldn't see it until they developed the electron microscopes. And some of the stuff I'm telling you about, they didn't discover until the 1990s, because this stuff is so small. But anyhow, inside the cell, you have this little skeleton of tubes and ropes, basically cables, and that gives the cell kind of a shape. And attached to all of that stuff, and we'll see this in a future picture, are little contractile proteins that actually give certain cells the ability to move. Okay? So what we have right here, where you have this cytoskeleton that goes throughout the whole inside, they have to attach to the cell membrane, and they attach to proteins that sit on the inside. So the anchor proteins allow the cytoskeleton to attach to the membrane. Recognition proteins, so the immune system recognizes the cell as self. We got this little guy called what? COVID?
Oh.
Right? Yeah, a little guy called COVID, which is a virus, which is an acellular infectious protein. It's not alive. It's just a little blob of either DNA or RNA. That's what a virus is. It has the ability to hook onto a cell membrane. It will inject its RNA and DNA, and the cell reproduces it. That's basically a virus. But on the surface of all of your cells, you have these little surface recognition, you have these little surface recognition proteins on the surface of every cell. So if I'm a white blood cell and I'm sneaking through the body and I'm checking to make sure I'm on the community watch system, and as I go through and I check and I say, okay, yeah, that's the right protein, he's a member of the body, right? But then I come over into that and I think, whoa, never seen that before. If he doesn't have the right surface protein, he is non-self. As a white blood cell, what am I going to do to him?
Eat him up.
I'm going to take him out. Do you follow me? So, first, you have anchor proteins, then you have recognition proteins, so that the white blood cells know if they are self or non-self. Got it? Thirdly, oh, by the way, some of the things that make viruses hard to detect is they have this little protein coat. I call it a blankie. Helps my students get the idea. And as these viruses come and they hook on, they pull this little blanket or protein over themselves, and it makes them harder to discover because they're cloaked. Sneaky little dogs. Okay, then you have receptors. They bind to specific extracellular molecules and hormones. So here you are, a little cell. Do you need sometimes groceries from outside to build? Yeah, so you have proteins in the surface of your cell, which basically will be able to grab what it needs from the outside and pull it in. Some of it is random, and some of it is like Amazon special order. You catching this? I mean, literally what can happen is because you have this bilayer like this, and here's the outside environment, the outer layer will reach out like this and just kind of randomly grab and pull in like this and force that in. This opens then, so you never lose homeostasis. One of those two is always closed. And as it shoves that down, whatever is in there gets wrapped around by a little part of the cell membrane, and it forms either a sac of waterish stuff, or if it's proteins or other things, then it forms another sac called a vesicle. And you're constantly bringing this stuff in or taking it out, and that's called vesicle trafficking. So, are you involved in trafficking? All the time. Either dumping stuff out or pulling stuff in. Those are the receptors. Carrier, they transport stuff across the cell membrane, and then the cell membrane, the certain protein types permit the passage of water through the cell membrane, because that's waterproof. So there's like a tube protein that goes all the way through, and it's normally closed. But sometimes what it will do is it will open up and it'll allow certain stuff in. for instance, and this is crazy, so you have these things called ions, like sodium ions, chlorine ions, potassium ions. For every kind of ion that is out in the fluid around the cells, for every kind of ion that you have, every cell basically has about 10,000 of these transport proteins that will, when you need, for instance, more sodium inside the cell, these sodium transport ion channels will open up and allow 2 to 3 million ions per second per cell per protein. And you've got 10,000 of them for each kind of ion. You say, whoa, 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 slow down. Do you see how complex this is? And it's all under auto-regulation. Your brain's not involved in that at all. Otherwise, you'd really be up at nights worrying about if your ion pump channels were working or not. Let's go back to this. So, the receptor proteins right here, they bind to specific stuff outside the cell to get molecules or hormones inside. You with me? And occasionally I'm going to just dump some heavier stuff on you so that you sit back and say, whoa, no way evolution. And this is where it gets a little bit heavier. Signal transduction. Don't worry about this. So here we are outside the cell. Do you see our cell membrane right there? You've heard of adrenaline? So adrenaline is a little molecule. It comes floating along in the bloodstream, comes out of the blood cells, or out of the blood vessels, goes to the extracellular fluid, and it binds to you gotta stop doing that. It binds to a protein receptor on the outside of the cell called the adrenergic receptor. That activates GDP, guanosine diphosphate, guanosine triphosphate, which breaks ATP, adenosine triphosphate, into adenosine monophosphate, which activates phenyl, keto, and then what happens is all of this. Simple? That's one receptor. So, Mr. Ed, do you see the engineering in here? And it's just, this is one of thousands. In the liver alone, you have more than 22,000 enzymatic systems. 22,000. And if too many of those go bad, then your liver is toast. Do you kind of get what we're talking about here, just briefly? So chemicals out here have to activate stuff inside the cell, so they bind to that receptor protein, and then you have this cascade mechanism. Boom, 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 boom. And the desired end result right here, and that ends up causing transcription, which is part of protein synthesis. This is just going on in all your cells all the time, even when you're sleeping. Hopefully you don't understand everything I just said, because if you do, I'll just give you an A in the course and we're done. But do you get the complexity of this, folks? Complex. Okay. Do you see what we're looking at here? Phospholipid, phospholipid. Okay. Now you don't have to listen to me.
Looks like a rug.
Looks like a rug. Thank you very much.
Not too simple.
Okay, how come this is not working? Pastor, what's the deal here now? It gets up to here and it disappears.
Oh, I see what you're saying, yeah.
No, it was working before you came. Oh, you got it, good.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Okay, let's see if we can do this.
Between the living machine.
Is this one the next up? According to your handout? The cell? I think it is. No, I don't want to do that. So the cell, that's next. Okay. So now what we're going to do is we're going to look at the cell briefly as a whole, and then we're going to look at the some of the subparts because we don't have time to go through all of it. We'll be here doing this for weeks. Okay. I've never seen it do this before.
You could just click. There you go.
Okay, anywhere, huh? Part of that's the cytoskeleton. That's the phospholipid bilayer. This is all taking place in your cell, and you'll understand more of this as we go along. Cytoskeleton again. Protein synthesis. RNA. Manufacturing of a tubule, part of the cytoskeleton. That's a motor protein moving stuff inside your cell along the cytoskeleton. And that's what it's carrying. Cytoskeleton to the nucleus. Protein synthesis. Another motor protein carrying its load. It's the Golgi apparatus, which we'll talk about. Yeah, you get kind of creepy feeling, don't you, knowing this is inside you? Yeah. So white blood cells sneaking outside of us, capillary. Do you see all this stuff that's taking place in that side there? Darwin made the statement, if it's ever determined that an organ is complex, my theory is in trouble. Did you catch that? What are organs made up of? 2 or more kinds of tissues. What are tissues made up of? The same kinds of cells. How complex is a cell? Way beyond what they ever thought an organ was. So if Darwin was alive today, being intellectually honest, he would have to say, my theory's in trouble. Okay, so major parts of the cell. We talked about the cell membrane, the cytoplasm, that's the goo in the nucleus. So 2 parts to the cytoplasm. The cytosol, that's the gooey stuff, and then it contains dozens of different kind of miniature organs. What is an organ? It's a group of stuff designed to do a specific function, like your heart, right? Designed to contract and pump blood. Well, as we got the electron microscopes busy and we started looking inside the cell, they found all of these different kinds of miniature organs that did specific functions. So, what is an organelle? It's basically a miniature organ that does a specific function. So we get smaller and smaller and more complex. So here's just a few of the cell organelles. So there's your cell membrane on the outside. All of this stuff in here, that would be the goo, that would be the cytosol, loaded with different kinds of structures called organelles, which they couldn't see until the electron microscope. So one of the first things that we're going to look at is this series of folded membranes that go back and forth. This is called the Endoplasmic Reticulum. And there's 2 different types, smooth and rough. These guys make lipids and they make proteins. Okay, really complex. But your cells are literally stuffed with stuff. And then we're gonna talk here. So here's your microtubules. There's intermediate filaments. We won't talk about the centrosomes. They're just too hard to try to explain. Not that you guys aren't bright, but it's really complex. We'll talk about chromatin. We'll talk about ribosomes, the Golgi apparatus. So enough to probably keep you confused. Okay, the endoplasmic reticulum. This is one of the organelles. It's a system of winding canals or tubes that extends from outside the nucleus all the way out to the cell membrane. And if I were to show you a photomicrograph from an electron microscope, you would see that literally your cells are stuffed with this, like there's no more room in them. And what do? Protein and fat synthesis and molecular transport. This is what it looks like. So you have here the nucleus, out here would be your cell membrane, and you have this series of folded membranes that actually were created by the cell membrane. And it actually forms these as the cell is developing. So the rough part, these little guys here, that makes proteins. The smooth guys out here make lipids. And when we get, not today, and probably in about 2 weeks, we'll talk about protein synthesis. Phenomenal, the complexity of it. Golgi apparatus. It takes the finished proteins, and so off the assembly line of the endoplasmic reticulum, at any given time, you are manufacturing north of 100,000 proteins per second per cell. Did you catch that? 100,000 proteins per second per cell within. And as they come off their finished assembly lines, what the Golgi apparatus will do is as these finished proteins come off the conveyor belt, it will look at them. If they're not perfect, it will correct them, quality control. It will pack all of the same kinds of protein inside a packing box called a vesicle, put a shipping label on it made of protein, and ship it wherever in the cell it needs to go, or if it needs to go to a distant cell, that sac goes to the surface, it gets kicked out, and it gets carried to the cell somewhere distant in the body where it needs to go. That will grab it, pull it in, and use it. It's like FedEx, except it's 24 hours a day, and you don't have to pay for shipping, and they never lose packages. Crazy. Let's pray. Father, what a marvel the human body is. What a marvel the cell. What a marvel even the organelles are, and they show your handiwork. Lord, help us to understand that as the creation, we can do better in worshiping you as the Creator. Give us opportunities this week, Lord, to share just your majesty and the most important thing that you've ever done for us, and that is the gift of eternal life. As we go now into the worship time together, may we worship you acceptably in spirit and in truth. We pray in your Son's name. Amen. Yes.
What is medicine?
I'm sorry?